Hey class, and welcome to part 3 video notes. So let's dive in to part 10.9. So transcription produces genetic messages in the form of RNA. So a lot of this that we're going to go over right now is just a review from what you've already learned in class. So here's an overview of transcription. So we have a strand of DNA, and those double helix, that double helix will separate. So those two strands are exposed. One strand, what we call the coding strand, is used as a pattern to produce an mRNA chain. So it's kind of like how you can trace a pattern. So we're taking that mRNA and we're like tracing that coding strand of DNA. That way we can use it to make a protein. So for mRNA, we already talked about how that U is replaced. And that we don't have U in DNA. What we haven't talked about yet is that RNA polymerase. Now this is an enzyme that's going to catalyze the reaction. Now the word catalyze, I'm sure you guys have probably already talked about in class, but it starts that reaction, it gets it going, and it keeps it going. So there's different stages to transcription. So the first part is initiation. That's where that RNA polymerase is going to bind to the promoter. The promoter is on DNA, and I'm not really worried about you knowing the word promoter and terminator all that much. Just be familiar with what they mean. So initiation, so that RNA polymerase, that enzyme that's going to catalyze this reaction, binds the promoter on DNA. The helix will unwind so that way transcription can start. Then we have elongation. That's where the RNA nucleotides are added to that growing chain. We have those nucleotides coming in from the cell, and we're adding them to that growing message. And then we have termination. RNA polymerase will reach a terminator or an end to the sequence, and then it will detach, and we can terminate transcription. So let's take a look at a picture. So here's our RNA polymerase. It looks kind of like a computer mouse. So in blue we have our DNA. So it unwinds. Now keep in mind this is just a picture that represents a really small segment. Normally it's a much longer segment. So our pink is our newly made RNA. Keep in mind it's single stranded. So you'll notice that instead of T here we have U, which we already talked about. So that T is replaced with U. So this polymerase is what's catalyzing the reaction. It's what's adding those RNA nucleotides onto that growing strand and making our message. So we have our RNA polymerase. It's going to bind to that promoter, that little green segment of DNA there. It's going to initiate the transcription, and it's going to continue to go. It's going to make the RNA message, and then when it hits the terminator, that red portion, it'll go, oh, time to stop, and then the RNA is completed. We have a completion of that message. So 10.11, transfer RNA molecules. So this is the first time we've talked about tRNA. So this is a really important thing when we talk about translation. So molecules match an amino acid to its corresponding mRNA codon. So tRNA is kind of like the interpreter. Remember how we talked about when we translate, we're going from one language to another language. We're taking those nucleotides and we're translating them to amino acids. Well, tRNA is that interpreter. It's what changes that language from one to another. So the amino acid is at an attachment site that allows the tRNA to carry a specific amino acid. So on this tRNA, there's a little portion where the amino acid will bind to it, and that tRNA will carry it around. An anticodon, which I'm sure you guys are so smart, you guys already know that it's going to be complementary to the codon triplet pair. It's going to allow that tRNA to bind to a specific mRNA codon, and it's going to be complementary in sequence. So in other words, A with U, G with C. So just like with mRNA, tRNA uses U instead of T. So 1012, ribosomes are what builds polypeptides. So it's what builds those proteins. Ribosomes are all over the cell. They're in the cytoplasm, they're in the rough ER, but right now we're floating around in the cytoplasm because remember, we leave the nucleus with our message and we go into the cytoplasm. So this is where our translation will occur. Ribosomes have two units, one small and one large. Each one of these subunits is composed of rRNA, ribosomal RNA, and proteins. These subunits are going to come together during translation, and they have binding sites for mRNA and tRNAs, and then it's going to allow that amino acid chain to form a protein. So here's this picture. This brown glob is our ribosome, and here's that section where that tRNA, that little green right here, can bind to the ribosome. Okay, so we see that mRNA going in and that growing polypeptide leaving. So that's our whole goal, is making that polypeptide. So here is a picture of a ribosome. Now this is definitely just a drawing because in real life it's going to look all crazy. So here's our larger subunit, it's the large one, and our small subunit. So this is where tRNA is going to bind in this portion and in this portion. And then here is where mRNA binds. So let's take a look. So here is where our tRNA has our growing polypeptides. So each one of these shapes represents an amino acid. Here tRNA comes in with a new amino acid that's next in line to be added to our growing chain. And then here's where our mRNA comes in. Our message comes in the ribosome to be translated. So you see how tRNA sits here and it kind of matches up with mRNA? That's because it's our interpreter. It's what's changing the language. Alright, so 1013. An initiation codon marks the start of the mRNA message. So initiation, so the beginning, brings together the components needed to begin the RNA synthesis. So this is going to take two steps. mRNA, our message, is going to bind to that smaller ribosomal subunit, remember at the bottom? And then the first tRNA binds to an mRNA at the start codon. What you do need to know is that the start codon will always read AUG. So when we talked about that chart of proteins in class on Thursday, that is going to read AUG. So that's always start. The first tRNA will have the anticodon AUAC. So see how they're complementary right there? The large ribosomal subunit will join the small one. It'll come on top, and it'll allow that ribosome to function properly and make that protein. So that first tRNA will occupy the P site. So the P site is part of the ribosome. So that P site holds the growing polypeptide chain. Now the A site is where that new next tRNA will come in. So look here again, you see that first site, that's the P site, where that peptide chain's already occurring, and then the A site here is where that next amino acid's going to come in. So here's the start of our genetic message, and the dark pink is what we're going to be using to make our protein. And here we have our terminator, or the end of the sequence. So our initiator, it's always MET. The amino acid MET is always what starts. So it's attached to our tRNA with a little hydrogen bond. Our anticodon and our start codon from the mRNA match up. Then my larger ribosomal subunit will come in, come on top, and then translation can occur. Now the P site is where that growing amino acid sequence is going to take place. That A site is where a new tRNA molecule is going to come in with another amino acid. So elongation, so this is our next step. That's what's going to add amino acids to the polypeptide chain all the way until we reach that stop codon. So in that worksheet you did, you found a stop codon, and that's what told you to stop translating. So elongation is the addition of amino acids to that growing protein or that polypeptide chain. So each cycle has 3 different steps. That codon recognition. So the next tRNA will bind to the mRNA at the A site. That's going to make a peptide bond formation. So those 2 new amino acids in the chain are going to bond, and they're going to attach to each other. Amino acids on the tRNA at the P site are then attached by a covalent bond to the amino acid at the tRNA at the A site. So it's a continuous process. So tRNA is going to come in at the A site, attach, and then that growing amino acid chain is at that P site. So translocation. tRNA is released from the P site, and then the ribosome moves tRNA from the A site into the P site. So this is going to take some explanation. You're going to have to find a person who owes you a favor or tell your dog all about these steps, so that way you can better understand it. Because if you can teach it, then you can understand it better. So elongation, so we're adding amino acids to that chain. So it continues all the way until we reach that stop codon, and then we have termination. So that's when that polypeptide is finished being made and it's released. The ribosome can separate large from small subunits, and then the mRNA is released. It can either be translated again to make more of that protein, that same protein, or if our cell is done with it and we have all the protein we need, then it's degraded. So this is a very, very important figure. I want you to go through this, think about it, talk about it, read it in your book, and make sure you really, really know this figure. So these are some questions I want you to take time to write down the answers to in your notes as you read these. If you don't know one of the questions, do your best to guess or write a question mark next to it, and we'll go over it in class. So now we get to talk about mutations. And so what I've seen in class is you guys really like to talk about mutations. So they can change the meaning of genes. So we're changing the language of our DNA. So it takes the nucleotide sequence, so our ACTG, our sequence of DNA, and it will change it. Now there's 2 different major types of mutations. There are base substitutions, where we replace one nucleotide with another, so like A with a G, and there's deletions or insertions. So that's when nucleotides are grouped into different codons. So base substitutions, the effect depends on whether there is amino acid change. So if you notice when looking at the chart, sometimes more than one triplet code will code for one amino acid. So it's kind of redundant. So if the change doesn't affect the protein being made, then we're fine. With deletions or insertions, that can possibly lead to significant changes in amino acid sequence, because we're completely deleting nucleotides, and that will change the sequence of codons, which will change the sequence of amino acids, which will change the protein. So that can have a really, really big effect, and it can cause a completely non-functional protein to be produced. So mutations can be spontaneous, so it's just an error that happens in DNA, because gotta think DNA is made all the time. We're always changing, or we're always taking DNA and reading it and replicating it. So that can happen by being spontaneous, or we can have a mutagen. So radiation, chemicals such as too much sunlight or smoking or other chemicals that can cause your DNA to mutate and give you mutations. So here is an example. So CTT to GAA, that's normal hemoglobin. Hemoglobin's in your blood. Mutant hemoglobin is where that A is changed, or where that T is changed to an A. That T is changed or I'm sorry, the A is changed, so that way this way we have sickle